So I'd like to thank the committee for enabling me to present this data. So I'm going to talk, this is a new approach for non-surgical sterilization that involves targeting the gonadal support cells. So I'll first give you some background, a little bit on fetal anti-Mullerian hormone, or Mullerian inhibiting substance, MIS, in the adult female male, and talk about a nanocomplex, which we have been using, and then preliminary data and hypothesis and on the experiment that we've recently done. And just to let you know, we do have an ongoing project as well as one that should start next month. So anti-Mullerian hormone has been sort of an orphan in terms of people using this in terms of controlling reproduction. It's been out there, and certainly everyone was taught about it relative to development. Okay, you start out with the mesonephric duct and the paramesonephric duct, and in the absence of testes, you make a female tract. But with testosterone from the testes and the Leydig cells specifically, and anti-Mullerian hormone specifically from the Sertoli cells, you induce the paramesonephric duct to essentially undergo atresia, and you support the development of the male. And this was where most people have left anti-Mullerian hormone, not thinking that it did much in the adults until around 1990. When research started to indicate that, yes, indeed, we have the production of this hormone, okay? And in the postnatal male, pre-puberty, it inhibits Leydig cell proliferation and thereby testosterone production, because you don't want testosterone produced before the animal actually hits puberty. And the anti-Mullerian hormone 2 receptors are actually found on prepubescent Sertoli and Leydig cells. And in the adult, the receptors are found on Sertoli cells, and recently people have indicated Leydig cells as well. The serum anti-Mullerian hormone actually seems to be reduced in relationship to androgen production. So once you start producing testosterone, there seems to be a fall in what you see in terms of the level of serum anti-Mullerian hormone in the male. But in the male, it's recently been shown by Anderson and his group that anti-Mullerian hormone instead now is essentially contributes to the semen. It's found in the semen as opposed to going outside and being found in the serum. And it does seem to be related to sperm numbers and quality. So we're beginning to find out that these cells do quite a bit in the adults. In the female, so anti-Mullerian hormone is secreted by granulosa cells in humans from week 36 of gestation to menopause. Okay, and in the dog, we know that the receptors on the repro organs are actually turned off at 5 weeks of gestation. So this is the question of, well, if you have these receptors on the organs during development, why doesn't, anti-Mullerian hormone then impact it in the adults? It turns out the receptors on the reproductive organs themselves seem to be shut down. All right. So as this morning Dr. Pepin already talked, that anti-Mullerian hormone in females inhibits recruitment and growth of primordial follicles. Okay, and without this, your reserve of primordial follicles is depleted much quicker. Okay, it decreases responsiveness to growing follicles to FSH, and it's been used in IVF in humans to indicate the number of follicles per follicular wave, in cattle as well. So, is the cow a good candidate for superovulation? And then the use of serum anti-Mullerian hormone to distinguish between ovaryectomized and intact female cats and dogs. And I will also say that there's a lot of evidence out there now that the granulosa cells and the Sertoli cells are not only involved in producing anti-Mullerian hormone, but other substances that probably, if not directly contribute to the control of meiosis and mitosis in both the male and the female. So these cells are very important in general to reproduction. And so I teach all of this to the veterinary students, and I had the thought early on that, well, gee, if you could get rid of these cells, if you took them out, you essentially have no means of reproduction. These cells also make hormones, so you essentially decrease hormonal production, you decrease the support of the developing germ cells, and you don't have any anti-Müllerian hormone. So my initial hypothesis was to selectively destroy all these cells with anti-Müllerian hormone receptors. It turns out, at least in humans, that the great preponderance of these receptors are on the gonads, right? There are some in humans in the adrenal gland, but compared to what you find in the gonads, it's minuscule. And then, according to the literature, a few here and there. Now, the mouse literature actually seems to indicate that there might be receptors in the brain, but we'll talk about that in a second. Anyway, so this was all very interesting, but I had no way to deal with this. I'm certainly not a person that can make vaccines or work with antibodies in that respect. And 3 years ago, Dr. Xiaobing Zhu called me up out of the blue and asked me if I had any use for a nanocomplex that he and his lab were developing in the Department of Biomedical Engineering at Tufts. And so the idea was this was a lipid, this was not metallic, a lipid nanoparticle, all right, that essentially could be guided by an antibody or small peptides, and it could either contain a drug, all right, or a cytotoxin, or actually have one attached. He had been using this method of carrying cytotoxin into antibody-guided, a way of antibody-guided means to target cancer cells. So this is what he had been using it for. And he said, oh, it was suggested to me to call up a repro person and see if you had any use for this. And I said, oh my gosh, yes, I do. I could kill those cells by using your technology. So we got together, and with who is now Dr. Christy Meadows, who was one of my students who was really big into immunohistochemistry, to decide how we might use his technology as a method to essentially gonadectomize mammals, right? So it turns out that because this is a lipid, so I mentioned that you might find receptors in the brain, that this won't pass the blood barrier. So if there are receptors in other species other than mice in the brain, this nanocomplex actually can't pass the blood barrier, much to Dr. Zhu's dismay, because he'd love it to be able to do this, all right? He has a huge library of lipids. He's done immense amounts of research on this. So he is essentially, this is his end of the research. Okay. So we needed initially, since we were going to target cats and dogs, to prove beyond a shadow of a doubt that these receptors actually occur in the gonads of cats and dogs, as we couldn't find any literature at the time that indicated this. So Dr. Meadows was actually very important in terms of getting this started. We had very little money, we're self-funded, and decided to use commercially available anti-Müllerian hormone receptor 2 that were on the market, all right? We used rat, cat, and dog testes and ovaries, and they were flash frozen and sliced with a cryostat. And then we used a primary antibody and a secondary antibody and vector red to essentially stain the receptors. And sure enough, here in this picture, this is a peripubertal cat testes. Dr. Meadows actually was working part-time in a clinic, and so she convinced them to donate their cat and dog reproductive, their gonads. And then if we look in the adult dog and the adult cat ovary, you can see the inner parts of all the circles are granulosa cells. Yes, you can see these. And the outer layers are actually theca cells that are taking up the stain. I have a student this summer who's actually doing similar work, and she's looking more specifically at age groups and using a different stain, and this is paraffin fixed, so that we can actually identify the cells more precisely, which was a problem with our fluorescence. The slices are thicker, and it's more difficult to tell exactly what cells you are staining. And so here, this is a female dog, and you can see the follicles, the granulosa cells are stained, all right, with the dark brown, and the theca cells around the outside. I have a pointer. And you can see on the right-hand slide, that's a secondary follicle with, again, a lot of staining. And if you look off in the periphery of that slide to the right, you can actually see primordials and primary oocytes. And again, the granulosa cells are taking up the stain. And so we were very happy that this essentially verified what we saw with the fluorescent stain. Here are peripubertal dog testes, and you can tell that this is prior to puberty in that they don't have a lumen in the seminiferous tubules. But look at all the cells that take up the stain. The potential here is really great if we weren't having some sort of strange cross-reactivity going on. This is the puppy with the other stain, and again, it verifies what we saw with the fluorescent stain. And this is the cat and male dog, seminiferous tubules, and again, they take up the stains. So we were very happy about all of this. Here's the rat ovaries and testes, and in this, you can again see, it looks like, to my eye at least, the theca cells actually take up the stain more intensely than the granulosa cells. And this is not surprising that anti-Müllerian hormone is part of a peregrine feedback system. So in other words, you need the latex cells to make androgens that the granulosa cell uses to make estrogen. So it's not surprising that there would be receptors and that there would be feedback. And then this is on the right, a rat testes. And there's some suggestion, although Dr. Pepin has suggested there may be cross-reactivity going on, that maybe even the sperm might have receptors for anti-Müllerian hormone. And since this is secreted in the semen itself, perhaps, there is a reason that anti-Müllerian hormone has a role. So what we learned was we confirmed that there were anti-Müllerian hormone 2 receptors in the male and female, adult rat, and peripubertal cats and dogs, and that the stain was indeed taken up by Sertoli, Leydig, and granulosa cells. The new information that we gained was that the theca cells in the follicles of these animals had anti-Müllerian hormone 2 receptors. And maybe the sperm within the seminiferous tubules may also have receptors. We also learned that commercial antibody could be used as part of the nano complex. All right? So we had the hypothesis, all right, that injecting the nanocomplex guided by a commercially available anti-Müllerian hormone 2 antibody, all right, so that it would be guided to the anti-Müllerian hormone 2 receptor, okay, carrying a cytotoxin, okay, could destroy the granulosa cells, the theca cells in the ovary, and the Sertoli and possibly Leydig cells in the testes. So this is sort of a diagram of how this could work. We used the cytotoxin saccharin, and this was preferred by Dr. Zhu because it is not effective alone in the bloodstream. So if this nanocomplex were to come apart in the bloodstream, all right, the saccharin would do nothing or very little. It has to be actually taken into the cell in order to be effective. So one of our next little projects, again self-funded, was to see if we injected this nanocomplex guided by an anti-Müllerian hormone antibody, would this work? Could we induce apoptosis? Because saccharin induces apoptosis in the cells in which it enters. Okay. So this was another set of preliminary data. So we had our nanocomplex with an commercially available anti-Müllerian hormone 2 antibody. We had male and female rats, only 10, and 4 of each of the male and the female were injected IV, either with saline, 2 nanomoles, 5 nanomoles, or 10 nanomoles of the nanocomplex. And then 1 male and 1 female were injected either intratesticularly or IP very close as we could estimate to the ovary itself. Dr. Zhu was concerned that whether the nanocomplex could get to these organs IV, right? And they were sacrificed after 24 hours, and we collected the gonads, right? And they were labeled for apoptosis with a tunnel stain, right? And fluorescent images were also obtained, and the data was analyzed and quantified, all right, using image J. And here are our results. So the red is tunnel. And yes, indeed, after 24 hours after injection in the rat, IV, we could see apoptosis in these cells, right? And the blue is just DAPI to show that we have live cells. And so our results were that it seemed more effective with this antibody in the males more than the females. And all of the males that were injected with more than 2 nanomoles showed a fair amount of labeling, and it was statistically significant compared to the 2 nanomoles. And the females injected IP was greater than all the other females in terms of the effect. So the local injection worked better with the female. And there was minimal effect in the saline-injected animals, as you can see here. So all you saw, you could see the DAPI, but there's nothing, right? So we were very, very excited, but it's such a small number. And it was only 24 hours, and we have no idea of safety, efficacy, and we needed to do another study, all right? And so well, here's what we learned. So the anti-Mullerian hormone receptor-guided nanocomplex, all right, can induce apoptosis within 24 hours, and that different routes of administration seem to be effective. And then we have all these other questions. So what specific architecture is impacted? What's disrupted? And do the effects last beyond 24 hours? Do we have something that can recover? Is there an effect on the health of the animal? We have no idea. All of this is a total black box. There's no information out there on this. And were there signs of decreased fertility after 30 days? So this was the next experiment. We had to beg the, it's the Clinical and Translational Institute at Tufts to fund us. And they did so as part of One Health, okay? A very, very small grant. But our hypothesis was that, yes, indeed, after 30 days, we should see a reduction, okay, in fertility, either in the architecture or in the case of the male, the sperm. So this was our study. I apologize for the small, but we had 16 animals this time, 8 males, 8 females, 4 were control, 4 were experimental, all right? And we used the same antibody that we had used in the 24-hour study. So it's commercially available. And the experimental animals were injected with 8.2 nanomoles IV. Now, this was less than we wanted. There appeared to be a miscommunication with the postdoc in Dr. Zhu's lab. But anyway, so it was a small amount, okay? And in the case of the IV injections in the experimental animals, we gave 0.1 mLs followed by 0.1 mLs of a sterile saline flush to make sure we got as much of the nanocomplex into circulation as possible. And then controls had 2.1 milliliter IV doses of sterile saline. So they were housed for 30 days. They were weighed twice a week. And the last 2 weeks of the 4-week experiment, the females, we did vaginal smears, all right, to see if we had indications of whether or not they were cycling. And after 30 days, they were all sacrificed by CO2. And the left and right gonads were collected and weighed. I actually took pictures of them. We decided sort of last minute to collect the epididymis and put them in sterile saline and look at sperm later on, because we were busy doing all of this, taking out the gonads and putting them in formaldehyde and measuring them, weighing them. So this was taking quite a bit of time. And then, so the gonads were in 10% formaldehyde for histological processing. And what we wanted to do was essentially stain them with H&E so that we could definitely see the architecture of the gonads. So We did not see any adverse effects on the general health of the animals, at least relative to weight, general activity, and whatnot. You can see the blue lines are the males, and the orange and red are the females. And yes, the males gain weight at a greater rate than the females. That's perfectly normal, but there was no difference, okay, between the treated group and the non-treated group. So we'll talk about the males first. Okay. Epididymal sperm, it turns out that actually rat sperm can sit at room temperature for 6 hours and still be viable. No wonder there's lots of rats around, right? Yes, okay. So, but what we did see was definitely an effect. We saw far less sperm in the treated animals than the non-treated. And considering that sperm can remain in the epididymis and whatnot, it takes 60 days to really clear out all the sperm out of the testicle, that this was very impressive. And here's a picture. On the left, you can see this was an animal that was treated, and on the right, here is the epididymal sperm of the non-treated animal. And I mean, it was obvious by eye that the treated animals had far less sperm. Okay. Now, comparison of seminiferous tubules was more daunting. I'm not a histologist, and I took these to pathology and actually had the pathologist look at the males and the females, and his comment was, whoa, these are not happy-looking gonads in the treated animals. And so he encouraged me to actually do some more look at these more carefully in terms, and here are some of the differences that we can see. So the saline controller on the left, the nanocomplex are on the right, and what we found was, and if you look and compare, not only are the edges irregular, it turns out that there were not normal sperm in the center. On the left, you can see this nice swirl. And so what we found, the abnormalities were atypical luminal contents, no sperm or undeveloped sperm, acellular material, immature sperm cells, disruption of the normal patterns. In other words, we would see, instead of seeing the nice spermatogony and then primaries and then essentially round spermatids elongated, you would see they would almost look mixed up, all right? So there was a statistical difference in terms of the number of normal seminiferous tubules that we could see out of 80. 78 out of 80 were normal. And in the experimental animals, only 24 out of 80 were normal. Okay, this is just higher magnification to show you what we saw. And so the seminiferous tubules on the right are definitely very different from the saline controls. So actually, Dr. Meadows did some more work and looked at the number of spermatogonia and primary spermatocytes, right, and found again there was a statistically significant difference. So now we didn't wipe out everything, but we're definitely on the right track of definitely decreasing these cells. Now in the females, all right, so here's vaginal aspirates, and what I found was that the animals that were treated were very confusing. Now, I could definitely tell what was going on with the normals, but you can see with the treated animals, which are N for nano, that I have a lot of question marks and A's. A means I saw all types of cells in them. I didn't see, the progression from parabasal to, eventually cornified. No, I saw a mix, and I have a picture of that. So here's what I saw in a lot of these treated animals. So I would see everything. I'd see white blood cells, cornified, intermediates, parabasal, and the parabasal were oftentimes vacuolated. Right? And I took these to people who did a lot more time looking at vaginal smears than I did normally, and they looked at these and said, well, I couldn't tell you what stage these are. What the heck is this? And when I took this to our reproductive endocrinologist, Dr. Bob Bridges, he looked at this and said, oh, I've seen this before. And I said, what? What was this? And he says, oh, we called them duds. And he said, we could never get animals that showed this type of vaginal smear to breed. We never got litters out of them. So this was also promising, far too my amazement, so to speak. Now, we haven't done as much detail on the female ovaries, but one of the things that we did see was that if you look at these at 2x, so essentially the same magnification, the treated animals have relatively larger ovaries. Okay, and it looks like that they have a lot of essentially edema going on, particularly in the central part of the ovary. All right, this is just a bigger magnification. They don't look like polycystic ovaries. I looked at a lot of pictures of those, and they don't. They tend to look like early senescence. So this is interesting in terms that I have no idea whether in this case you are seeing something would be ongoing if we looked at this farther out, or whether it's part of recovery. That's something that we really need to do studies at a longer time to determine. This was just a very rough count, and what we could see that there was an increase in the treated animals in the number of CLs. Now, I didn't differentiate between CLs that might be normal versus those which were undergoing atrophy. And the same thing with the tertiary. There were more tertiaries, but again, I'm not an expert at identifying those follicles that are undergoing atresia. And so this will take further looking at this data to see what's going on. But in general, it looks very promising. Now, this has just led to a lot more questions, all right? And so one of the things is, would there be differences in the receptors relative to the estrous cycle? And that's something we actually have some slides on, and we need to look at that, all right? We did look at that 2 summers ago. And how effective is this nanocomplex relative to the age? And just by eye, it looks like there's a lot of staining pre-pubertal, and whether or not this would make a difference. We have an ongoing study with young pigs where we actually injected it a week after birth, and so we'll have some data on some of that. And what is the relationship of dose to the effectiveness? Can complete sterility be achieved, and will it be permanent? And are the serum hormone levels impacted? We would have loved to have looked at hormone levels, but my blood samples were all hemolyzed. You can't look at hormones with hemolyzed samples. Okay, so the nanocomplex seems to disrupt gonadal architecture, okay, seminiferous tubules, a decreased number of spermatogonia and primary spermatocytes, increased size of the treated ovaries, increased numbers of tertiary or antral follicles and corpora lutea, can disrupt the normal cyclicity, can reduce the numbers of epididymal sperm, and all of these changes would result in a decreased fertility. So, future work, we're going to analyze those theca and granulosa cells relative to the estrous cycle in the rats. The Mars Foundation has given us a small pilot grant to actually look at this in avian species. It turns out that the exotic veterinarians at the vet school got wind of this and they said, oh, we want it. If it works, this could eliminate the problem we have of treating avian species where we cannot do surgical gonadectomies. But perhaps if we can reduce the hormones that are produced, we can have a great effect. So anyway, they're very interested. And we have an ongoing pig project where we've actually given the nanocomplex IM as well as IT. Those animals are over 3.5 months old right now. And I can tell you, I go down and help weigh them and take pictures of their rear ends every 2 weeks, that definitely the testicles of the treated animals, both IT and IM, I can see by eye, all right, are decreased. And they, according to the herdsperson who doesn't know who's who, but he says, you know, some of those guys there's atrophy going on. So this looks very, very promising, all right? So we're also working with an immunologist, because one of the questions that has come up is the safety factor. Again, if you're going to make an IM injection, what happens if it's IM into a person? all right? And we're working with immunologists who say that there is probably a good way to work on the antibodies such that it is more species specific, or at least not specific to humans, so that they're convinced that there can be ways to do this so that it makes it safe, all right? And we're proposing, we're looking for money for efficacy and safety and fertility studies in rats, cats, and dogs. That's our future work. So I'd just like to acknowledge everybody who's helped here. Definitely Dr. Zhu. Without him, this would never have happened. Dr. Christy Meadows, the veterinary students, Eric and Sarah, the Tufts Tech Transfer Office, the patent people. They have been very enthusiastic and very helpful about this. The CTSI people who gave us the funding, all right, and then the Department of Biomedical Sciences for all of their support. So with that, I'd like to conclude and thank you all.